Hello, everyone. I'm Jordan Zambrana, a biologist with the EPA Indoor Environments Division. I'm pleased to host the latest IAQ Science webinar titled The Impact of Relative Humidity on Indoor Chemistry and Microbiology. Before I introduce our speaker, there are a few points I need to mention. This webinar is being recorded and will be available on the EPA website at a later date. If you have a question during today's presentation, you can type it in at the Q&A box on the right-hand side of your screen at any time. The questions will be read to the speaker at the end of the discussion. If you have any technical questions or comments, you can submit those inquiries through the QA box as well. We look forward to hearing from you. I also want to reiterate that the content of the webinar represents the research and opinion of our speaker and not necessarily those of the U.S. Environmental Protection Agency. So with that said, I'd like to introduce Dr. Karen Dannemiller. She is an assistant professor at The Ohio State University with a joint appointment in civil, environmental, and geodetic engineering, as well as environmental health sciences. She directs the Indoor Environmental Quality Research Group, which studies microbes and chemicals in the indoor environment. One of the areas under investigation by this group is the association of indoor microbes and chemicals with childhood asthma. So Karen, thank you again for speaking today, and I'll hand it over to you to begin the presentation.
Great. Thank you so much for your kind introduction. I'm really excited to be here today to talk to you about our recent research efforts. So as you said, my name is Karen Dannemiller. I'm an assistant professor at Ohio State University. And in the Indoor Environmental Quality Research Group, we study the indoor microbiome and indoor chemical exposures and how those relate to childhood asthma. So we spend 90% of our time indoors, and this is especially true now during the ongoing pandemic. And the indoor environment is actually fairly complicated in terms of the mixture of chemicals that are present, as well as a diverse community of microorganisms. So in the Indoor Environmental Quality Research Group, we study how the indoor environment itself contributes to what chemicals are present, what microbes are present, the interactions between those chemicals and microbes, and ultimately how these impact human exposure. When we think about exposure in the indoor environment, much human exposure originates from floor dust. And in fact, about 60% of the particulate matter in the indoor environment comes from resuspension from the floor. So anytime you walk into a room or move around a room, you resuspend particulate matter up out of the floor, that gets into the air, and that can ultimately result in exposure, not only to that particular matter itself, but any microbes or chemicals that might be present there as well. And you can imagine a lot of situations where you might get more exposure if people are moving around the room more rapidly. There's more human activity. You can get more microbes resuspended into the air. And when we think about these exposures, this is especially problematic when we have an unhealthy environment, such as in the presence of mold. Mold in housing is a huge problem in our society, and in fact, it's estimated to cost about $22.4 billion every year. And it's usually not as pretty as this piece of artwork shown here. In fact, it's associated with about 16% of the cost associated with allergic rhinitis, acute bronchitis, as well as asthma. This is not only an economic problem, but this definitely impacts the people who suffer from these different conditions. And probably most of us know at least one person who has one of these conditions. And a lot of these have both a genetic or medical component, but also an environmental exposure component that can, these in-home exposures can actually make these conditions worse. Even though we know mold in housing it's harmful, we still don't have a quantitative test to measure it. In fact, if you take a look at this recent article by Mark Mendel and Rachel Adams, you can see different ways that we might measure mold in the environment across the x-axis on the bottom, and across the y-axis you can see odds ratio, or the association between the indicator and negative health effects. A lot of our quantitative microbial measurement methods shown on the left don't have very strong associations with our health outcomes. However, if you look on the right, measurements of moisture or simple observations of dampness and mold or detection of moldy odor have very strong associations with health outcomes. it seems like we should be able to do better to determine a more quantitative microbial measurement method that could be used in homes to determine which homes are healthy and which homes are not. That could do better than these more subjective measures of visual inspection or moldy odor. And to do this, we need to really fundamentally understand what is happening in these homes when they become damp, when we have excess moisture in these homes, and what happens to the indoor microbial communities and to the chemicals that you might be exposed to in that particular indoor environment. That brings me to the outline for my talk today, which focuses on two parts. The first part is focusing on what happens when you have excess moisture in a home to the microbes in that indoor environment. So if you think about our system here shown on the right, the indoor environmental parameter that we're going to really focus on is moisture, because we know that that's associated with known human health effects. In the second part of my talk, I'm going to talk about excess moisture in housing and how that might influence indoor chemistry. I'm also going to talk a little bit about how the microbes and the chemicals within the home might interact with each other, which could ultimately result in changes to human exposure. So for part one, we wanted to ask the question, how does moisture in housing influence the indoor microbial communities? There's been a lot of studies on materials such as drywall, where we looked at what happens when drywall gets wet, what types of microbes tend to grow there. But we really wanted to focus on carpet and flooring materials, and in particular, floor dust. The reason we wanted to focus on this is because, as I mentioned before, we get a lot of resuspension of this material up out of the floor, and that is a large contributor to human exposure. So we wanted to ask, can elevated moisture in a home change those microbial communities? And in particular, we were focused on situations where maybe you do not have condensed water. Maybe you just have elevated relative humidity levels. We also focused on carpet because it's such a prevalent flooring material in homes, especially post-World War II in the United States. If you look at this graph, you can see the percent of carpeted rooms over time. And today, carpet still makes up about 50% of the US flooring market. So it's still a really common material used in homes. Additionally, carpet is also a really important reservoir of human exposure to dust. And so we actually had a workshop in summer of 2019 where we got together 40 experts who study all different aspects of carpeting in homes and ask the question, what does putting a carpet in a home, how does it change the indoor microbiology and indoor chemistry? This was a really exciting workshop. We brought together people from governmental agencies, including the US EPA, as well as carpet manufacturers in the cleaning industry, and academics from all different disciplines, all sorts of different disciplines. And I definitely encourage you to take a look at this paper if you want to learn more. But one of the main findings of this workshop was that carpet is such an important reservoir because not only does it retain more of the particulate matter and material in the indoor environment, but it also resuspends it at a higher rate compared to solid surface flooring under most typical loading conditions. So it's a really important source of human exposure to dust. In addition to being an important source of human exposure, we also know that the relative humidity in carpet can be elevated above that in the room air. So if you look at this graph on the right, this was a study where they measured the relative humidity in the room air and compared it to the relative humidity level in the carpet. And you can see that in many cases, the relative humidity in the carpet was actually higher than that of the room air. This is true for many surfaces. Measuring the relative humidity in the center of the room may be very different than the relative humidity right above a surface. And so in this case, you can actually potentially have more moisture available to the microbes that are present in this environment, depending on a lot of different conditions in the home, such as temperature gradients. There's a lot that can happen when carpet gets wet. We might see an increase in dust mites. We might see changes in indoor chemistry, like we're going to talk about in part 2. There might be changes to bacteria, but we're really going to focus on the potential for fungal growth here. So the first question that we wanted to ask is relative humidity in the air sufficient to support microbial growth in carpet? So we wanted to say, even if you don't have condensed water, can elevated relative humidity actually support that microbial growth and ultimately impact human exposure? So to look at this, we collected carpet and dust from a home. Here you can see a before and after picture on the right. So we found a home that wanted to replace their carpet for aesthetic reasons. They didn't have any history of mold damage or water damage. And we selected an area of carpet that was in a high traffic area, cut out the carpet, and collected a matched dust sample from the home as well. We then took that back to the lab and exposed it to elevated relative humidity conditions and measured the microbial growth in terms of the rate, the microbial function occurring, and some phthalate degradation, which I'll get to in part 2. When we bring that carpet back to the lab, we cut it into 10 by 10 centimeter coupons. We apply the dust and we embed it with a modified ASTM method. ASTM does actually have a method for just about everything, including embedding dust in carpet, and it works quite well to actually roll this heavy pipe over the carpet. Once the dust is embedded into the carpet from the matched home, we place the carpet coupon in a temperature and relative humidity controlled chamber. We place different carpet coupons at different relative humidity levels, and we can then study the resulting microbial growth. To look at that microbial growth, we use DNA-based techniques. So we will take the dust from the carpet sample, we will extract the DNA, and we will do DNA sequencing, and then use bioinformatics tools to measure the microbes that are actually in that home. This will result in hundreds of thousands of DNA reads per sample, which will give us taxonomic identification or a list of the microbes who are there, quantification if combined with qPCR, as well as an overall measure of diversity, so we can get a really comprehensive picture of how the microbial communities change as they're exposed to these different relative humidity conditions. The first thing that we looked at in this study is how the dust itself actually absorbs this moisture. So for this, we looked at water activity. Water activity is a measure of water availability that's commonly used in the food industry. So for instance, the food industry might use water activity measurements of things like granola bars to see if you would expect microbial growth, because it's such a good measure of if that water is actually available to microbes. Water activity is equivalent to the equilibrium relative humidity. So you can think of it like a relative humidity of 95% is in equilibrium with a water activity of 0.95 is a good way to think about it. So the first thing we looked at on the graph on the left, we have the relative humidity on the x-axis and the water activity on the y-axis. And we saw that after 1 day and 1 week, the water activity equilibrated very quickly with the relative humidity in the jar. And we also saw visual changes in the dust. So if you look on the right at figure B, we have dust incubated for 1 day at these different relative humidity levels and then after 1 week. And we knew that we must be onto something because after 1 week, we actually saw visible fungal growth on the dust itself. And we said it looks like we're probably going to get some growth showing up in these samples. And indeed we did. We were actually able to see that microbial growth occurred in the dust starting at about 80% relative humidity level. So if you look at the top left graph, we have fungal concentration on the y-axis and the original dust and different relative humidity conditions across the x-axis. And you can see that as relative humidity increased, we got an increase in fungal concentration. On the graph to the right, we also saw an increase in bacterial growth, but only really at 100% relative humidity levels. We didn't really see an increase in growth at the lower relative humidity levels. And this makes sense because fungi are known to be able to grow at lower moisture conditions as compared to bacteria. So the story here is really largely about the fungi unless you have a really, really high relative humidity level. We also studied the relative humidity conditions over time at 100% and 85% equilibrium relative humidity in the jar. And you can see that this growth continued over time. We had actually originally intended to grow the 100% relative humidity samples out to 6 weeks, but we had such extensive visible fungal growth after 3 weeks that we decided to end that experiment and that we had shown that this growth definitely does occur. So overall, we were able to see growth at these different relative humidity conditions. We also wanted to see if the microbes in these samples might be limited by nutrient availability, and it does not look like that is the case. So we measured different nutrients that are needed to support this microbial growth. So on this table, you can see different nutrients down the left-hand side of this table, then the dissolvable amount that was actually present in the dust, and on the right, the estimated amount needed to support pretty extensive growth, 1 week of growth at 100% equilibrium relative humidity, and none of these nutrients would be limiting in actually preventing this growth from occurring. So it really is moisture that's the limiting factor for growth to occur in the system that we're looking at here. Next, we wanted to also see if this growth would actually contribute to human aerosol exposure. So this is a rough resuspension model where we looked at the concentration that would be expected in the air for both fungi and bacteria. And in the orange at the bottom, you can see the background level that you would expect. And then in the blue and the red, you can see the additional exposure you would expect at these different relative humidity conditions. So it is possible that this growth in the dust can contribute substantially to human aerosol exposure. Now, one thing I want to note about this first study is that no one leaves their house at, say, 90% relative humidity for exactly a week, or at least I hope no one does. That would definitely cause problems. So we wanted to start to make this a little bit more realistic. So for instance, the US EPA recommends that the relative humidity in your home be between 30 and 50% and definitely below 60%. And those are really good recommendations based on what we saw in our data. And that's probably the conditions that occur in most healthy homes. However, there are conditions that occur where the relative humidity is elevated for short periods of time. For instance, you can have a showering event, and when a shower happens, your relative humidity might raise up to 100% for, say, 20 minutes in the air, but it actually can remain elevated above 80% for up to 6 hours after that. So if you have a room where someone's showering in the morning and someone showering at night, you could have 12 hours a day where the relative humidity at the surface is elevated above 80%. You can think of other conditions where relative humidity might become elevated, such as a hot, humid summer day with windows open after a cooking event. If you have water intrusion problems, such as water leaks, these could all cause excess moisture in the home. But we wanted to ask the question, what if this moisture is transient? It doesn't stay at these elevated levels. So to answer this question, we conducted our next study where we collected carpet samples from 19 homes around the state of Ohio to look at how variations in relative humidity might affect fungal growth in carpet. We really wanted to answer two questions with this. One is, do we still see this growth if the relative humidity is only elevated for a short period of time? And two, can we actually model this growth? As engineers, we wanted to ask the question, can we quantitatively know how much growth we would expect to occur under any given set of varying relative humidity conditions? So for this study, we took our carpet and dust, the same system I talked about before, back to the lab, and we incubated it at varying relative humidity conditions. So for these samples, we would start them at 50% relative humidity, and then we would elevate the relative humidity to either 85 or 100% for 6, 12, 18, or 24 hours a day. You can see the graph on the right, which shows the theoretical variations in relative humidity that we attempted to achieve, and then also the data logger at the bottom that showed what actually happened in these jars as we changed the relative humidity conditions. After doing this to model this growth, we applied the time of wetness framework. And this was a framework that was originally developed to model microbial growth on drywall. And what this framework says is that the time of wetness, here you can see across the x-axis, or the percent of time that the samples spend above a growth threshold of 80% relative humidity, is proportional to the relative growth rate, here shown on the y-axis as R over K. So the more time you spend above this critical growth threshold, the faster the microbes are going to grow. And there's actually 4 models within this framework. The first is the standard model, shown here as the solid blue line. And that says that these are linearly proportional. So the more time you have at elevated relative humidity, you should get a linear equivalent in terms of the increase in growth rate. Next, in the orange, is the activation limited growth model. And this model says that there's some delay in restarting fungal growth after dry conditions end. So as soon as you have wet conditions occur, you wait a little bit and then fungal growth will restart. The deactivation limited growth model is shown here in green, and this basically says that there's a delay in ending of fungal growth. So as soon as you go from wet conditions to dry conditions, it takes a little while for the growth to slow down. And finally, in red, the 2 activation model is actually a combination of the activation limited and the deactivation limited growth model. So we wanted to see in our system which of these models would apply most closely. So the data from this study was very exciting. We did, in fact, see that even short periods of time at elevated relative humidity conditions can sustain fungal growth. So in this case, we were looking at up to 6 hours per day at either 85% or 100% relative humidity. Not only that, but we were able to model this fairly well using the time of wetness framework. So here you can see on the left our time of wetness framework graph for 85% relative humidity. In the middle is 100%, and on the right is a reminder of what this graph looks like. It appears like our data most closely fits the 2 activation model. However, we didn't take any data points between 0 and 6 hours a day to confirm that. So that's something that could be done in future work. But it does look like if we know about how much time the materials are spending above this critical growth threshold for relative humidity, we can actually predict how much fungal growth would actually occur. In addition to this, we had some really interesting findings about what species were actually present. So this is a principal coordinate plot where principal component plot where similar microbial communities cluster together. And so these were all the samples in the study from all the different sites. The sites here are shown in different colors that were incubated at the different moisture conditions. And the darker the dot, the more moisture that the sample was exposed to. And the reason that I'm showing you this graph is actually because it's kind of a mess. So if you've been trying to find a pattern, I'm really sorry. But I wanted to show this actually because there's not too much of a pattern. What we had actually hoped to find with this graph was that there was a darker grouping of dots on one side and a lighter grouping of dots on the other side. And that would tell us we had this clear species-specific signature of excess moisture in homes. And that's not actually the case. Here you can actually see that a lot of these samples clustered much more closely by site. So what this says is actually the site-specific effects actually dominate any moisture signature in the microbial species present. So for instance, this shows that each home is actually pretty unique. Each home has its own unique microbial signature, and excess moisture doesn't actually change that signature enough to be detectable. This explains a little bit why it's been so challenging to develop an indoor indicator of microbial growth in homes that is based on species, because each home is so unique. It's really hard to find a specific signal to indicate if you have a moisture problem based on the species that are present. So we were really excited about this particular finding. We wanted to follow up on this as well to look a little bit at the microbial function. So we got really excited now that we know these microbes are growing in our system. We wanted to ask, what are they actually doing? So for this, we conducted a study to look at the gene expression of microbes at 50%, 85%, and 100% relative humidity levels. So this is a plot here, once again, where you can see that similar groups of gene expression samples clustered together. These samples were all from the same home. and just incubated under these different relative humidity conditions. And the interesting thing is that overall you see pretty similar gene expression profiles at 50%, 85% shown in yellow and blue. But these are very different from the samples incubated under the high moisture conditions, the 100% relative humidity conditions. And not only that, but these samples were actually very different from each other. So when you get to these high moisture conditions, you actually have quite a bit of variability in terms of what the microbes are actually doing. They can do a lot of different things. They now have enough moisture available to support more and different metabolic activity. So this is a KEGG ontology plot that actually shows the different genes that were expressed in these samples that I just showed you. This is probably my favorite GIF. I'm still waiting for it to go viral on social media, so look for it on your feed. But at the top left, you can see the different water activity levels that were measured. So the one equivalent to 50% relative humidity, 85% relative humidity, and 100% relative humidity. At 50% relative humidity, we really just have those core metabolic processes that are occurring. Carbohydrate metabolism is complete. Lipid metabolism is complete. But really, it's just those fundamental processes really necessary for life that are happening. At 85% relative humidity, we see a little bit more on the outside, but then at 100% relative humidity, you actually see all of these secondary metabolic processes around the outside light up. And that is really interesting because that tends to be where the allergens are and the virulence factors and the mycotoxins. So it's very likely based on this data that the samples exposed to these elevated relative humidity conditions are producing things that might tend to cause more health effects, such as more allergens. We wanted to check to see if that was actually true. So on the left, you can see a violin plot where we looked at the gene expression of known fungal allergens. And we did actually find that overall, more allergenic genes were statistically significantly upregulated at 100% relative humidity compared to 50%, 85%. So we did see more allergen gene expression at the higher moisture condition. We also wanted to confirm this with a protein-based assay by measuring the allergens directly themselves. So this had to be done in a specific species with high water activity requirements based on the limitations of the assay. But we were able to also show that at least for this species, we did indeed find more allergen potency per spore at higher moisture conditions. And so this confirmed, at least for the species, what we suspected, that as your moisture increases, you do get more allergen production per spore. This really also begs the question of, is a spore in terms of health effects, or does it really matter where that spore was grown and what conditions that spore was exposed to during growth? So this is something that I think is really important, and once again goes along with what we found earlier, that it is really challenging to find an indicator of microbial growth in homes because it really matters where these spores came from. We also confirmed this again in another study. So here we looked at Alt-A1, which is an allergen of Alternaria alternata, and we grew these samples at different relative humidity conditions and measured this allergen specifically, and once again saw that at increased equilibrium relative humidity conditions, we see that the moisture, that the more moisture you have present, the more allergen is actually produced in these samples. In this study, we also looked at a lot of other interesting factors. We actually wanted to compare how the moisture availability compared to dust, compared to fiber type, in terms of determining how much growth you actually get. We once again confirmed that the amount of moisture present was the most important factor in determining how much microbial growth you'll get in a carpet and dust sample. The dust loading was also important. So we saw that at increased dust loading, you could get additional microbial growth, but it wasn't as important in the moisture, especially at most typical dust loading levels that you'd find in a home. Finally, we did see that different carpet fiber types did also influence how much microbial growth you got, but it wasn't as important as the moisture or the dust. In this study, we also took some really exciting pictures of the growth at these different relative humidity conditions. So these are 5 different relative humidity conditions: 50%, 85%, 90%, 95%, and 100%. And these show SEM images of nylon carpet fibers with dust present. You can see at 50% that we don't see any evidence of fungal growth. We just mostly see dust particles present on the nylon carpet fibers. At 85% and 90% relative humidity, though, we do see evidence of hyphal extension. So we're starting to see that microbial growth occur, similarly to what we saw in the previous data I showed you. Finally, at 95 and 100%, we see pretty extensive microbial growth. We see the presence of fruiting bodies, and we really see that this aligns very well with what we saw in our previous study, that as your relative humidity increases, you do get an increase in the amount of growth present. We also took a Z-stat confocal image. So here you can see the nylon carpet fiber in gray and the fungal growth around this fiber in blue. And you can kind of see how the fungal growth actually wraps around this carpet fiber and uses it as a scaffold. It seems to be that in most cases, the fungi are probably using the dust as a source of food, but it looks like they might wrap around the fibers depending on how they're oriented. So I'd like to provide a summary of part 1. Overall, we found that elevated moisture in a home was sufficient to support microbial growth and function in dust. You don't actually need condensed water. Elevated relative humidity is sufficient to support this growth. And this has really important implications because we need to make sure that we are continuing to control moisture in homes and pay attention to the surfaces where moisture might be present. So we need to think about, where we're putting carpet in a home, make sure it's not in an area where it could be exposed to excess dampness that would then support this growth. So we also looked at some of the microbial function in dust, and we determined that you can actually get increased allergen production at these elevated moisture conditions. So once again, it's really important to control the relative humidity within homes, especially at surfaces, and especially in the carpet and dust, because this is such an important source of human exposure due to resuspension. Next, I'm moving on to part 2. So for part 2, we're going to look at moisture again. And this time we're going to study how this might change exposure to chemicals in the indoor environment. How does this moisture actually influence indoor chemistry? And I'm going to focus here on 2 specific projects. The first is to look at degradation of compounds in dust. If we get excess moisture, can we get chemistry that's occurring in the dust? The second part, I'm going to talk about an ongoing study where we're looking at release of volatile organic compounds and microbial volatile organic compounds from building materials, once again, under elevated moisture conditions. So for the first study that I'm going to talk about, we're using the same system I talked about before, but this time we wanted to ask more specifically, what can this microbial function do? We showed that this microbial function is occurring in the dust, and we wanted to know, can it degrade chemicals that are present in the dust, like phthalates? We wanted to look at phthalate esters because they are so common in the indoor environment. They're used as plasticizers to make plastic flexible, and they're not actually covalently bound to the polymers, so they leach out pretty readily. They're actually found in high concentrations in house dust. In some of the homes with the highest levels, you can find levels up to about 1 milligram per gram. These are also endocrine disrupting compounds of public health interest, so they can act kind of like hormones in your body and cause potential health effects. Finally, we wanted to start with these compounds because we know that they can be degraded by microbes in other systems, such as wastewater and soil. And we wanted to ask the question, can microbes in house dust also degrade these phthalates? On the right, you can see here one common phthalate. This is DEHP. This is probably one of the most common phthalates that is used and that we're going to talk about in this study. So for this study, we conducted 2 experiments, once again, in the same system I've been talking about before, and these were all done at elevated relative humidity conditions. So the first is what I'm referring to as the natural experiment, a sort of quote unquote natural, because this was just looking at the phthalates that were already present in the dust sample that we collected to use for this experiment. So we looked at these 9 different phthalates. And then we also conducted a spiked experiment where we spiked samples with deuterated DEHP, which we could track over time, and we could also track the byproducts of its degradation. So the first thing we did is we looked at the results of our natural experiment. We incubated these samples over the course of about 3 weeks, here shown on the x-axis of these graphs, and we looked at these 9 different phthalates and their concentrations over time. On these graphs, you can show the low molecular weight phthalates on the left side and the high molecular weight phthalates on the right side. And what we observed was that we saw preferential degradation in the dust of these high molecular weight phthalates. This was really interesting because we actually had expected the opposite to occur. In other systems, like wastewater, we commonly see preferential degradation of the low molecular weight phthalates. So we weren't sure exactly why this occurred, but I believe we did figure it out over the course of the study. So I'll get to that in a bit. However, at the big picture level, we were also really excited to see that we did see phthalate degradation in dust. So this indicated that this degradation was occurring, and we wanted to study a little bit more how. From this initial experiment, we couldn't tell if this degradation was due to the microbes in the dust or to other chemical reactions that were occurring. But we were able to look at that in the next experiment. So for the spiked experiment, we took 4 different dust samples and we spiked them with deuterated DEHP, and we measured the percentage that was lost over time. We had sterile controls included in this, and so we were actually able to determine how much of the loss was due entirely to the chemical processes that were occurring, or the abiotic processes, and how much was due entirely to the microbial processing in the dust due to that microbial degradation. So the chemical loss here is shown in blue, and the microbial loss here is shown in orange. And you can actually see that over time, we saw both abiotic and biotic losses in all 4 samples of house dust. So this was particularly exciting that this loss did actually occur. And this is one of the first studies that we're aware of to actually look at this chemical and microbial interaction in this house dust system. We did know that there was actually quite a bit of variability in each of these samples in terms of how much degradation actually occurred due to both of these processes. And this is an incredibly complicated system. We have hundreds of chemicals, hundreds of microbes who are present potentially in each of these samples, and it's really difficult to know exactly which interactions are occurring. So overall, we were just excited to show that this does happen, and this is really just the tip of the iceberg to learn more about what's actually going on in these samples. We looked at some of the degradation byproducts. So the pathway that we were expecting to happen is that DEHP, our phthalate, degrades to MEHP, which then degrades to phthalic acid. So we measured MEHP, our first product, after a week, and phthalic acid after a week under all of our different conditions. And we saw an increase in these products as time increased. Additionally, we saw more MEHP produced after 1 week at 100% relative humidity levels. We also saw this change over time. So if you look at figure D, you can see that we actually saw changes in MEHP, which is both a byproduct and something that then gets further degraded into phthalic acid. And we saw the amount of phthalic acid also increase over time. Sometimes people ask me, so this sounds great. Maybe we should just increase the relative humidity in our homes to degrade all of our phthalates. And that's not a good idea for a couple reasons. The first reason is that obviously increasing the relative humidity is going to cause mold growth and cause unhealthy conditions. So that would not be a healthy thing to occur. The second thing is, even if you did want to degrade these phthalates, the byproducts are arguably more harmful than the parent compounds. So formation of these byproducts is actually additionally potentially more harmful. But we were able to show that these byproducts do actually form under these different conditions. Next, for this study, we wanted to look at the microbial function that ultimately resulted in the formation of these byproducts. We looked at the gene expression. And it was really interesting. I won't go specifically through these graphs, but we did find genes expressed in our system that are known to be associated with phthalate degradation pathways in other systems. And the really interesting thing is that the gene expression pathways that we identified are the ones that tend to be associated with preferential degradation of the high molecular weight, the larger phthalate compounds. So this actually was very similar to the data we showed before, where we had seen that preferential degradation of the high molecular weight compounds. It's hard to know, because this was such a small study, if this occurs just in this system or potentially in other systems as well. And there might be a lot of variability here in other systems. But it's really interesting to know that we were able to identify a lot of the microbial function that was occurring and potentially associated with this phthalate degradation occurring in the house dust. So once again, to summarize the study, we were able to show that the microbes in the home could interact with the chemicals that were present in the dust, and that at elevated relative humidity conditions, the microbes could actually degrade the phthalates, and the phthalates also degraded through other chemical reactions that were occurring that were not associated with the microbes. This did result, in this case, in the formation of byproducts that are also potentially harmful. And like I said before, this is really just the tip of the iceberg. There could be so many interactions occurring in the house dust system between these microbes and the different chemicals present. So this is a really great, exciting area of research where there's really a lot left to discover. All right, so now I wanted to move on to volatile emissions. And so before we were talking about the compounds in the dust, now I want to talk about the compounds that are emitted into the air. So I noted before that moldy odor is consistently and highly associated with health outcomes when I showed you this graph. So it's one of those things that is the most strongly, the factor that's most strongly associated with these health outcomes. And yet we still don't have a quantitative measurement technique to know which odors are most strongly associated with this and why. So we need to understand a little bit more about which compounds are emitted. So I'm going to talk a little bit about volatile organic compounds, which are compounds released into the air and those that you can smell. This is work from an ongoing project. So this is led by Rachel Adams at UC Berkeley, and we're also working with Pavel Mishtel at University of Texas at Austin, and students Sarah Haynes and Emma Hall are also working on this project. And the goal is really to understand how 3 different factors, moisture availability, geographic location, and material type, can impact 2 things: the microbial growth that's occurring, as well as the microbial volatile organic compound production, or the production of these VOCs that come from microbes. To do this, we're using a similar setup that I've been talking about with the carpet and the dust embedded into it. And this time we have the opportunity to use this really exciting instrument called a PTR TOF-MS. It can detect over 1,000 different VOCs that are released into the air. So we'll put our samples into the incubator, into these chambers shown here on the left, under these different relative humidity controlled conditions, and they'll go into the instrument where these compounds can then be measured and measured very accurately and actually in real time. So this is actually a game changer in terms of understanding which compounds are actually coming from these samples. This is a little bit what the instrument looks like. It's a pretty big instrument connected to all these sample jars. And additionally, there's actually been improvements in this technology where with some of the newer instruments, we can get 10 to 100 times thousands higher sensitivity than the previous generation and really get an incredibly comprehensive picture of what chemicals are actually released in this particular system. So for our initial results, I said we wanted to look at microbial growth. And one of the things that we wanted to look at was microbial growth in different building materials. So for this graph, on the left, you can see carpet. On the right, you can see drywall. On the x-axis is different relative humidity levels. And on the y-axis is fungal concentration. And one thing that we found to be really interesting is that here we see that the growth in carpet occurs at a lower relative humidity level than the growth on drywall. This has really important implications, both for the microbial concentration, showing that you would actually expect to get more microbial growth earlier in carpet at some of these lower sort of elevated relative humidity conditions like 80%, 85%, that might even be more common to occur in homes. Compared to drywall, where we really saw this growth just at the higher relative humidity conditions. This also has important implications for indoor chemistry, where you might actually expect more microbial VOC emissions to be coming from the carpet and dust samples than you would from, say, a drywall sample, just because they're more likely to grow at these lower relative humidity conditions. Once again, we're still talking about relative humidity conditions that are fairly elevated, so these relative humidity conditions above about 80%. So we were able to measure the VOCs that come from these building materials directly, and also those that come from microbes. So this graph shows some of the different VOCs that we measured. We have an emission factor on the y-axis and different relative humidity conditions across the x-axis. And here we're looking at sterile carpet on the top and carpet with dust and microbes embedded at the bottom. And one thing that you'll note is initially looking at these profiles, they actually look somewhat similar. There are some differences. But in this study, it was interesting because we found that even though we had off-gassed our carpet samples for months before using them in this study, most of the volatile organic compounds actually still came directly from the building materials. So you do have this much larger VOC emissions from the materials themselves. However, we did see indications that some of these emissions do potentially come from microbes as well. So we did see potentially microbial specific MVOC emissions. Some of the possible MVOCs that were emitted included monoterpenes, camphor, and dimethyl sulfide. And so these have been identified as potential MVOCs in previous studies, and in our study were also potentially associated with these non-sterile samples compared to the sterile samples. The interesting thing about these VOCs is even though they're in lower concentrations than the other VOCs being emitted, they can potentially produce odor. And so these may be some of the VOCs that are commonly associated with those microbial odors that you might smell in the indoor environment in, say, a damp or musty house. They do have interesting potential to contribute to this indoor chemistry that's occurring. And some of these microbial emissions may actually react with ozone for really interesting secondary chemistry that could potentially be occurring. So really, the VOCs in the environment, even though they do seem to largely come from the building materials, there is a microbial contribution to the chemistry in the indoor environment, especially at these elevated relative humidity conditions that we've been studying here. So to wrap up part two, we were able to demonstrate that microbes can actually degrade phthalate esters in house dust with sufficient moisture. And like I've said before, this is really just the tip of the iceberg. There's a lot of really interesting chemistry that can occur potentially in the indoor environment. There are so many chemicals present and so many different microbes that there are really a huge number of interactions that can actually be occurring. And this is just the first one that we've looked at. So a lot of really exciting stuff I believe will be discovered in the future about that. The moisture itself impacts indoor chemistry really differently based on the building material. So we saw that in drywall compared to carpet, we were able to get more microbial growth at earlier at lower relative humidity levels, even though they were still elevated. So this means that we might also potentially have more interesting microbial chemistry occurring typically in homes in carpet and dust systems compared to drywall. Finally, moisture has both direct and indirect influence on indoor chemistry. So we saw a lot of these interactions that occurred just because of the chemicals present in different chemical reactions that occurred. And we also saw this indirect influence on indoor chemistry. There's really a lot of exciting future work that can be done, especially with the instrument that we've been using for this recent study. We're hoping that we can maybe start looking for things like VOC signatures that might be consistent, that can indicate moisture in homes. And this we're actually at the point now where we can get probably the most comprehensive picture we've ever had in terms of the microbial communities that are present in these samples, but also their function and also their chemical emissions and the chemicals associated with these. So this is a really exciting time to be in this area of science and looking at what's ahead. So for my overall summary, we talked about in part one the elevated relative humidity, and especially even just elevated relative humidity, not even condensed water, is sufficient to support that microbial growth and function in house dust. This really indicated that it's so important to follow the recommendation of the US EPA to keep relative humidity in homes between 30 and 50%, and definitely below 60%, to prevent that adverse growth from occurring. It's important to think too about a home that the relative humidity in the center of the room might be different than the relative humidity on a surface. And that's something that we need to continue to think about going forward. We also saw interesting indoor chemistry that occurred through both abiotic and biotic processes in the home. So we saw that the microbes were able to degrade the chemicals, but we also had abiotic chemical reactions occurring too. And there's so much going on with the interaction between chemicals and microbes. It's a really exciting place to be. This has really important implications for the design of many environments. And you can think about all the different places that might benefit from this knowledge. This includes houses, offices, hospitals, school buildings, all of our buildings on Earth, and even potentially beyond Earth, such as spacecraft, where we can think about the interesting interactions that may occur potentially in these systems. So with that, I have so many people to thank for this work. Of course, the funding agencies, National Science Foundation, National Institutes of Health, as well as the Alfred P. Sloan Foundation, who funded a lot of this work. I especially want to note the chem team from the Alfred P. Sloan, who's funded through the Alfred P. Sloan Foundation, Rachel Adams, Pavel Mischel, and Sarah Haynes, and Emma Hall. As well as all of the other colleagues who collaborated on these different projects. It was fantastic to get all of their input. And I owe so much thanks to my students. I particularly want to call out for some of the data shown here, Sarah Haynes, Ashley Bope, Nig Nastasi, who contributed a lot to these different projects and led some of these projects that I've shown here. I'm also extremely grateful to the study participants, those people who took the time to fill out our surveys and call us up and donated their carpet and dust samples. Definitely tremendously appreciate their efforts to this study. And with that, it's a jungle out there, and I'm happy to take any questions.
Great. Fantastic presentation. Thank you again, Karen, for joining us today and for that really fantastic presentation. Thank you to all who joined us online and for submitting your questions. For announcements of future IAQ Science webinars, please visit our IAQ website, and we will begin with the questions now. And again, we are certainly not hurting for questions. We have plenty. So here is our first question. Recently, in an effort to reduce the risk of microbial growth in buildings, ASHRAE established a hard limit of 60 degrees Fahrenheit, 10 degrees Celsius, dew point humidity limit during unoccupied hours as well as occupied hours. That is in ASHRAE standard 62.1. Based on your work, do you think that this is an appropriate limit, or should it be higher or lower to limit the risk of high humidity in carpets and on cool indoor surfaces?
Thank you. That's a really great question. I would like to look a little bit more specifically at that standard before I comment directly on it, but I do want to commend ASHRAE for putting out a standard for unoccupied buildings. I think it's a really common problem. You think about schools, for instance, who close down over the summer, and commonly they'll have microbial growth that occurs because they have inconsistent heating and cooling. And it can definitely be a big problem. So I think we do need better standards for occupied and unoccupied conditions to prevent this growth from occurring under those unoccupied conditions. So I would say, in terms of the study that I've been looking at, for me, it seems to be the relative humidity that's really critical here to control and make sure it is below that kind of, like, if you're below that 60% threshold, you should be okay. And also considering, the surfaces, you want to think about the center of the room, you want to think about the surfaces as well. But that's really a good measure of that moisture availability in the dust. So if you think about dusty areas, they're going to be in equilibrium with that relative humidity. They're going to, their water activity is going to equilibrate. And that's going to be a really good measure of whether sufficient moisture is available to those microbes or not.
Great, thank you. And so a lot of the themes that came through that you were discussing were kind of that while moisture is very important in starting and continuing microbial growth from a bacterial and fungal perspective, when you tried to characterize the species, you found that actually site-specific kind of differences were more important really than purely just whether the moisture availability or not. So kind of what other factors did you see as or do you like posit could be driving these species diversity indoors? And is this in your mind kind of more evidence of an indoor microbiome separate from just whether there's moisture or not?
That is a great question. There are so many things that contribute to the microbes in our home. And I think a lot of that is what makes everyone's home so unique and have such a unique microbial signature. Things like ventilation rates and occupancy are really important. Whether or not you have pets, such as dogs or cats or other pets in your home, the geographic location of the home, the different species can vary. Geographically, the outdoor species can come in. Even different practices that you might have in your home are going to change the microbes that are present. So really, each home has this unique microbial signature that comes from all of these different places. And that's why I think it's been such a challenge, because when the moisture is available, they change in such different ways. You know, they're still very, very similar to those different microbial communities. The second part of that question is whether some of those influences could potentially also contribute to other health effects. And I believe the answer is yes. I don't think we have necessarily any highly conclusive evidence yet, but there is some really interesting evidence about exposures to things like dogs, or animals. We saw the emergence of the hygiene hypothesis, which basically says our homes might be too clean, and we might be missing exposure to critical microbes. And a lot of that work was done looking at animal exposures, which may potentially be beneficial. So we're still, and there's a lot of great work that came out of Susan Lynch's group actually identifying specific microbes that might result in that, and a lot of other great work that's been done. So I think a lot of these other factors can actually influence the microbes present in our home. One way I like to think about it a little bit is there's sort of two hypotheses out there in terms of microbial exposures. There's like a good guys hypothesis, which we need this exposure to good microbes. And then there's a bad guys hypothesis, which is we need to make sure we're not exposed to harmful microbes. If you're thinking about the indoor environment, probably the vast majority of microbes are neither. They're probably neutral, and you have a small amount that's beneficial, a small amount that's harmful. And in this case, I was talking more about the harmful ones, the bad guys, the bad guys, but the good guys are there too. And I think there's a lot of really exciting work being done and to be done in that area to learn more about actually both sides, which ones are harmful, which ones are beneficial.
Great, thank you. And so now I have a couple of questions about the specific carpet experiments that you conducted. Some maybe about kind of specific characteristics of the carpets which you used. So did you sample or include dust from area rugs? What were the CO2 levels during your experiments? Go ahead.
Yes. So these experiments were done on wall-to-wall carpeting, and carpet actually varies pretty dramatically. So here I'm using the term carpet, but it actually can mean sort of a wide variety of materials. In these experiments, we were only using wall-to-wall carpeting. We tended to focus on nylon because we were using a lot of older carpets. Today, polyester is a more common material. So if we were going to get like a newer carpet, we'd probably choose polyester as a different material. As I said, the material did seem to have some influence on what grew. So for instance, we used a wool carpet, and in the SEM images, we actually saw evidence of tunneling the fungal hyphae actually tunneling into the scales on the wool, which could potentially be using that in as a source of food, basically, potentially. So the carpet material itself can vary, but it looks like it has a relatively small influence on what actually happens. There is some influence there, but it's not huge. You can look up the paper by Nick Nastasi as the first author in Building an Environment. We have a lot of data in there about the different carpet materials and how that can impact the ultimate growth. In terms of the CO2 level, we're very cognizant that if you do have elevated CO2 conditions in your incubation chambers, that can cause problems in terms of fungal growth. So we did monitor the CO2 conditions and just made sure it stayed below a reasonable level. I think I'd have to go back to double check, but I believe in almost all cases it was below, I would say 600 ppm at the top, but we made sure that it was not becoming elevated because we know that that can cause issues as well.
Great. And one more question, were any of the carpets stain resistant or treated with something like Scotch Guard?
Yeah, so that's a really great question. In terms of the older carpets, we were typically collecting these from homes, and a lot of the carpets had been in the home for over 10 years. And so no one, there wasn't really receipts of who bought the carpet and exactly what it was. So it was not definitely known if there were some of these treatments in the carpets. I would assume in most cases, yes, because a lot of carpets do tend to have these types of treatments. But we did not look specifically at these treatments and what they did. So I think that is definitely a future direction where we could look at that a little bit more.
Okay. And would you suspect the carpet results that you had would be similar to interior lined HVAC vents, where there is dust from use and elevated humidity as well, but not direct water impact?
That is a great question, and that's one that I actually get a lot. So you think about your ducts, and you have these dust deposits on the ducts that are just sitting there. And I do believe that if you have something like a whole house humidification system where you're getting high humidity in the ductwork, that's going to cause a problem for that dust as well, because it's pretty similar dust. The dust is a little bit different from the carpet just in terms of its features. The dust in the ductwork tends to be more linty, where the dust from the carpet is sort of a mixture of like lint and more like sand-like material. But I would suspect that you are going to have a similar response. Microbes are everywhere in our homes. They come from us, they come from outside, they're in the air, and they're going to be in the ductwork too. And I would suspect that if you do have elevated moisture condition in the ductwork, you probably are going to end up with similar problems. I have not specifically looked at dust from ductwork though, but I would suspect that there would be a similar result.
Okay. The next question is, did you look at fungal mycotoxins in any of the samples? And what, if you did, what was the relationship between fungal growth on surface and the amount of mycotoxin in the air?
That is a really great question. So we did not specifically measure the mycotoxins in the dust. We did see that there was upregulation of genes associated with mycotoxin production at these elevated relative humidity conditions. So when I think of mycotoxins, I also think of things that are non-volatile, typically by definition. So I would expect them to remain largely in the dust if they were produced, but we didn't actually measure them specifically. We did see suggestive evidence that they are probably produced at these elevated relative humidity conditions though, but we don't have that measurement to confirm that. But that is a really interesting question.
And so this next question asks kind of, what is your opinion on products and carpets really specifically embedded with antimicrobial agents? Is this something you would recommend on flooring, carpet, or rugs?
I think when we think about antimicrobial agents, we need to be really careful because you do want to prevent exposure to the microbes, but you also need to be aware of what chemicals you might be being exposed to, especially if you have small children who are going to be really close to the carpet, crawling around on the carpet. So generally, I would not like to see additional extra chemicals added to these systems that might potentially be associated with health effects down the road. Generally, for a carpet, if you can keep it clean and keep the moisture away, you're going to be okay. And in fact, a lot of people actually think that I'm very anti-carpet, and I'm actually not. I do think that there are situations where carpet can be beneficial. It does have some good properties in terms of sound dampening, prevention and cushioning of falls. It's soft and comfortable. People like it. I do think, though, that we do need to be really careful about where we put it. We don't want to be putting carpets in bathrooms, for instance. That is not a place that you want to be putting a carpet. And wherever you put it, you also need to be committed to maintenance. So you need to be vacuuming it on a regular basis, and you need to make sure that it's not going to become a damp area and cause problems. So rather than application of chemical treatment, say antimicrobials, I would instead say that you need to keep it clean and keep it dry, and then you're probably going to be okay.
Great, thank you. That actually segues very nice into the next question is, are there any ways to make carpet less conducive for biological proliferation? So what do you recommend in that perspective then? And I think that you kind of answered that with regards to just keep it clean and keep it dry, but please feel free to add.
Sure, I'd love to add one thing actually, and that came out of the carpet workshop that we ran in 2019. And this was a really great experience. I so much enjoyed meeting everyone and hearing their diverse perspectives on this issue. And one thing that came out of that is it seems like it could be a really important environmental health goal to design carpets with properties intended to improve environmental health. And right now, that's not what a lot of people think about. It's maybe what I think about if I was going to go buy a carpet, but a lot of people, they're more interested in the color and what it looks like and how it feels. But there are ways, the carpet industry is incredibly smart, and they have some really great technologies. And we could work to improve how these carpets are actually designed to promote better environmental health, such as reduced resuspension, as well as, talking about cleaning, when you buy a carpet, how should you clean this? How often should you clean it? What do you need to do? And I think some of that it also needs to come in the form of consumer education. So I think it's two places, thinking about the manufacturers who can produce this and promote this idea that you can have a healthier carpet, and also the consumers to say, this is something that I want. When I buy a carpet, this is what I look for, and I'm going to choose the carpet that has better environmental health properties. So I think we should think about when we're designing carpets, how we're actually constructing them and how we can do things like reduce that resuspension rate, make them easier to clean in terms of dirt removal. Some carpets are designed to actually look clean even though they're not. And I think that can be problematic. You might not see the dirt, but it might still be there. And so I think that's something that we can continue to think about in the future of how can we actually design a carpet that might actually be healthier.
Great. So we have a question about the relationship between relative humidity in air and the moisture on or near surfaces. Like what physical chemical models did you use or could you point us to in understanding that relationship?
Sure. So I would look a little bit into, just read up a little bit on water activity. So the water activity is actually equivalent to the partial pressure of the dust over partial pressure of the water. And I would do a little bit of reading about that to understand that equilibrium. So a lot of the understanding of water activity, though, is done pretty much at equilibrium, where you have a chamber, for instance, and you just let it sit until everything comes to equilibrium. The indoor environment is also dynamic. So we have these changing relative humidity conditions, changing temperatures, things like that. So there's definitely a lot going on. But if you're looking to learn a little bit more about that, I would just look up some information on what is known about water activity.
Okay. I'm seeing a couple of different questions here about the fact that there's a benefit to a limited amount of relative humidity, and that there likely needs to be kind of a balance in humidity levels. What the question here, I guess, is do you recommend that there be a lower limit, a lower bound of a humidity level, and if so, how do you feel about that?
Yes, I think that the EPA's recommendation of humidity between 30 and 50% and definitely below 60% is a really good recommendation level. So what I'm looking at are the problems that occur at the high end of relative humidity, but at the lower end you can have other problems, such as, you may have been in a house where your throat feels really dry, your nose feels really dry, it's just too dry, your skin feels really dry, and that can be problematic. As well. So in addition, we also see that some viruses may be more viable at these lower relative humidity levels, these very low relative humidity levels. So I think it's important that we do have a lower bound on relative humidity. One thing that people ask me too is sort of what can I do about this? And I often recommend to people, you can buy a low cost relative humidity meter, for like $10 to $20. It's really not that expensive, and just measure what the relative humidity is in your home and keep an eye on it. You don't want it to be too dry, you don't want it to be too damp, and you just want to keep it in that happy medium.
Great, great. And we have another question here about, I guess, specific types of carpet material do you recommend? So say nylon versus olefin or wool?
Yes. So in terms of the actual carpet material, it didn't contribute that much to actually what type of growth occurred. The wool carpet was interesting because it looked like there was actually an interaction more with the fiber, where previously it was more with it appeared to be more with the dust particles. So generally, I don't have a specific recommendation in terms of carpet fibers. As I mentioned, most of the carpet fiber out there now is polyester, which does have some advantages in terms of it tends to be more naturally stain resistant, and so may have less coatings on it. Depends on the specific product though. So I don't have a specific recommendation. I would say generally get what you like. It does look like there might be less resuspension from looped carpet compared to cut tuft. So it may be a good option to go with a loop style carpet if you like it. But overall, I think just commit to keeping it clean and keeping it dry are the most important factors. And then otherwise, get what you like. I guess another thing about resuspension is a shorter tuft also probably results in less resuspension. So you might consider a shorter carpet.
Great. Yeah, that was really great that you went into the resuspension, because the next question is kind of, I think earlier on in your presentation, you referenced that 60% of exposure occurs from resuspension, and people were curious about a reference for that because that it was higher than some had previously observed.
Yes, and so that's under occupied conditions. So if you look at the literature out there, there are conditions where people are looking at unoccupied environments. So that reference was more towards occupied environments. So if you have people in the room, they're moving around, they're resuspending. And that's actually really when we care about the amount in the air. In terms of a vacant room, it's usually less of a concern of what's going on there. You might have someone walk in, but pretty much as soon as they walk in, they're resuspending that material out of the floor. So the reference that I was referring to occupied conditions, and you may find lower numbers in the literature, especially for unoccupied conditions.
Great, great. So our next block of questions are more concerned with kind of your future plans and future subjects of study. So the first would be, are you interested in studying growth in newly constructed homes where construction moisture can be responsible for excess humidity?
Yeah, I think that would be really interesting to look at that. I think anywhere that you have the potential for excess moisture, there's the potential for growth to occur. I think one interesting thing that could be done would be to apply the time of wetness framework that we used in our study to see if you can predict actually how much microbial growth you would get in specific situations. And that could also be used to then try to limit that growth. So you sort of know when you're going to run into a problem, and you can try to make sure that your moisture stays below that specific level. So I think that would be really interesting to really look at this in any system.
Mm-hmm. Absolutely, and towards that second part of your presentation, you really started to discuss the contribution of MVOCs to the secondary chemistry reactions. And just as another common household activity of cleaning, are you interested in studying kind of the reactions between microbes and household cleaning chemicals?
Yeah, I think that pretty much any chemical we're putting into the environment has the potential to interact with microbes. You can look, there are definitely papers out there about, disinfection kinetics that occur and how these cleaning agents might interact with the microbes. And that's an interesting one because you're applying these cleaning products which oftentimes are meant to kill the microbes. So I think that we also want to start to look at more of these compounds and how they might interact with porous surfaces, which is really challenging. A lot of studies are done on flat surfaces, like, for instance, stainless steel, and looking at disinfection that occurs there. I think working with carpet and these porous surfaces is really challenging. It's something that we've worked really hard at to be able to do well. And but I think it's important to understand how these products might also work on these types of surfaces, especially because they are so common in our indoor environment.
Great. Let's see here, looking for another question. Oh, yes. So did the backing of the carpet contribute any to the different results in fungal or microbial growth? So the one question was, did any of the carpets have jute backing, which is a backing I'm not familiar with, or any different organic medium different than, say, nylon?
Yeah, so we did look at some with jute. We did most of them were like flow through backings. We didn't specifically study the backing, and most of what we believe was happening was kind of in that kind of carpet matrix where the dust tended to sit in the middle of the fibers. I could see in the environment definitely some differences that could happen if, say, you have a rubber backing, which would be more common in like a commercial type material, that might potentially change what's going on in terms of the moisture. So in terms of the backing question, I think it's really interesting, but that is something that's to be determined.
All right, so I think that we have asked we have reached our limit for our questions are asked, the questions that were there. So I think at this time we will bring the presentation to a close, and I just want to thank you very much, Karen, for your time and agreeing to attend, and thank you very much.
Yeah, thank you so much for having me. I really enjoyed being here. It was really great to talk with you all. If anybody in the audience wants to reach out, I'm always interested to talk with people, hear what else they're up to. But it was really great being here, and thank you so much for organizing this and for inviting me.
Great, thank you.